Hello and welcome to the varvis corpus satellite entitled Sharpening Our Tools for the Diagnosis of Mosaic Disorders. In this session, our speakers will highlight the specific challenges that physicians encounter while handling mosaic diseases and explain in detail how a suitable assay was established and validated. Before we jump right in, let me briefly introduce to you who we are. My name is Yvonne Kassmann. I'm Product Manager at Nimbus Medical Technologies. Nimbus is a software development company located in the north of Germany, right at the Baltic Sea. We are a medical device manufacturer, which means that we operate and develop our products according to international medical device standards. Our product varvis is a cloud-based CE-label clinical decision support system specifically designed to aid in the diagnosis of patients. It's a complete solution for all clinical NGS applications, ranging from small panels up to whole exome sequencing, cancer diagnostics, and even applications like liquid biopsy. The session today consists of 3 talks. We will start with a clinical perspective presented by Professor Martin Zenker from the University Hospital Magdeburg, who will talk about the challenge of phenotypic heterogeneity and genetic diagnosis in mosaic disorders. The second part is presented by Dr. Ben Liesfeld from Nimbus Medical Technologies, talking about reconciling regulation and innovation with standardized software. Finally, we have Dr. Danny Schanze, also from the University Hospital of Magdeburg, who talk about matching analytical performance and clinical utility, where he will guide you through the process of establishing and validating the suitable assay in the lab. Finally, we will have some time for questions and answers. So please feel free to ask all the questions you have in mind. Let's jump right in with the first talk. For this, I would like to introduce to you all Professor Dr. Martin Zenker, who gained his medical education and experience mostly at the University Hospital of Erlangen-Nuremberg. He's a specialist in pediatrics, neonatology, and human genetics. Since 2009, he's the director of the Institute for Human Genetics at the University Hospital Magdeburg. His research focus is on congenital disorders of physical and mental development. And he's an internationally recognized expert of diseases related to the RAS signaling pathway, especially Noonan syndrome and neurofibromatosis, and their relationship to cognitive deficits, leukemia, and tumors. So today he will talk about the challenge of phenotypic heterogeneity and genetic diagnosis in mosaic disorders. Enjoy.
Okay. Mosaic disorders are a particular challenge because of their phenotypic heterogeneity and also regarding diagnosis. I want first to talk about the phenotypic heterogeneity. We usually consider mosaicism if, for example, the phenotype shows a regional or asymmetric distribution, if linear or patchy pigmentary anomalies are present, or if the phenotypic expression is unusually mild for the given disease. But actually, we have to consider mosaicism in every case where you have no other explanation for the phenotype and negative results are obtained from blood cell testing. Mosaic mutations may be variants that do also occur as constitutional mutations. In that case, the phenotype corresponds to the constitutional mutated phenotype and may present with attenuated features or regional expression. But mosaicism can be also caused by variants that are lethal as constitutional mutations, and these disorders are only seen as mosaic disorders, and the phenotype has no corresponding constitutional mutated phenotype, so to say an independent mosaic disorder. And this includes, for example, large-scale chromosome abnormalities, which I would say cause embryopathic phenotypes with multiple congenital anomalies. But the other type of mutations that are lethal in the germline are variants in oncogenes. And these cause a tissue dysplasia phenotype. In that case, you see lesions of tissue dysplasia where the mutation is. And I want to focus on this type of mosaicism. But I want to show you also some examples for the other types of mosaicism. First of all, mosaicism that occurs as constitutional mutations. For example, morbus Darrier mosaic, shown in the left image, and a segmental distribution of neurofibromatosis type 1 on the right side. In Cornelia de Lange syndrome, mosaicism has been observed multiple times, and it may be associated with an attenuated phenotype, and typically the mosaic mutation is shown better in fibroblasts or buccal cells than in blood. Mosaic trisomy 8 is one example of multiple examples with large-scale chromosome abnormalities that do only occur as mosaics. And in this case, you see a multiple congenital anomaly syndrome like in other chromosomal disorders. In the case of the variants in tumor genes, which are lethal as dominant mutations like PIK3CA mutations, it depends on the timing of the mutation, whether the phenotype has a segmental or multi-segmental distribution, or in the case of a late embryonic mutation, it may occur just as a singular mosaic lesion. So the determinants of phenotypic variability in germline mutation disorders or constitutional mutation disorders and mosaic disorders include, of course, the genotype and other non-genotype related variability. But in the case of mosaic disorders, the spatial expansion of the mutated cells or tissues, the timing of the mutation, and the affected cell or tissue type give us 3 more dimensions of variability. Among the oncogenic pathways that are involved in mosaicism and mosaic disorders, RAS, MAP kinase signaling pathway and the P3 kinase ACT pathways are one of the most important pathways, among others that also have mosaic disorders. And CLOVES syndrome, which stands for congenital lipomatous overgrowth, vascular malformations, epidermal nevi, skeletal anomalies, is one of the prototypic disorder of mosaicism, with typically classical oncogenic PIK3CA mutation occurring in the mosaic, and you can find it in the lesional tissue in these patients. The PIK3CA related overgrowth, PROS mosaic spectrum includes CLOVES syndrome and other complex phenotypes like fibroadipose, hyperplasia, MCAP syndrome, and others. And the elements of these are vascular malformations that may be capillary, venous, or lymphatic, epidermal nevi, lipomas or lipomatosis of tissue, macrodactyly, cortical dysplasia of the brain, and also tumor formation. Proteus syndrome is a classical mosaic disorder with ACT mutations, and it consists of cerebriform connective tissue nevi, epidermal nevi, vascular malformations, lipomatous overgrowth. So you see a lot of overlap with PROS and also tumor formation. In the case of the RAS MAP kinase pathway, Schimmelpenning syndrome is a complex disorder with systematized nevus sebaceous and other lesions that occur through HRAS or KRAS mutations in the mosaic state. And NRAS mutations or BRAF mutations as mosaics can cause giant melanozytic nevi or neurocutaneous melanosis. Oculoectodermal syndrome is another RAS mosaic disorder with multiple anomalies that consist in epibulbar dermatitis, aplasia cutis congenita, skin lesions and also bony lesions. And in this case of this rare disorder, we and others have shown that specific KRAS mutations are responsible for this syndrome, and consistently the mutations could not be demonstrated in the blood cells, but only in the affected tissue. The entire rasopathy mosaic spectrum consists of these phenotypes I mentioned before and others, and the elements are epidermal nevi, specifically nevus sebaceous, a keratinocytic nevi, melanocytic nevi in the case of NRAS, BRAF, vascular malformations, mostly of the high flow type, non-ossifying fibromas and giant cell tumors of the bones, choristomas, and also tumors. And there are different other cutaneous mosaic rasopathies I will not mention in detail. In all these cases, the mosaicism can be systematized with multiple lesions dispersed over the entire body, but also a single lesion can have the same or virtually the same mutation. And all these elements we see in the complex phenotypes may also occur as isolated lesions like epidermal nevi, melanocytic nevi, isolated vascular malformations, fibromas of the bone, or giant cell tumors. And in the case of giant cell lesions of the jaw and also non-ossifying fibromas of the bone, more recently the involvement of the MAP kinase pathway as mosaic lesions has been shown. Similarly, in the PI3 kinase ACT mosaic spectrum, isolated phenotypes may be venous or lymphatic malformations, cortical dysplasias of the brain, lipomas, or isolated macrodactyly, as shown in the next slide. In the case of these lesions, you usually find PIK3CA mutations, the classical oncogenic mutations in bone or adipose tissue. In this case, it was an allele frequency of 20%. Or cystic lymphatic malformations of the neck can have PIK3CA mutations. This brings me to vascular malformations, which can tell us a strong lesson of mosaic disorders. Vascular genesis and angiogenesis are very complex, tightly regulated processes of development, and they involve the pathways we know already, a combination of RAS, MAP kinase, and PI3 kinase ACT signaling. And you can see here that almost all known vascular malformation phenotypes can be plotted on components of these two pathways. And the mutations in the case of vascular malformations may include primary somatic mosaicism, as mentioned before, and this is true for isolated arteriovenous malformations, venous malformations, lymphatic malformations, and the complex syndromes I mentioned before, which include vascular malformations as one phenotypic feature. In the case of primary somatic mosaicism, an activating mutation of a signaling molecule leads to an increased signal flow. Vascular malformations, however, may also occur with germline mutations and as somatic second hit. And this is true for some of the familial vascular malformation syndrome, like familial capillary arterial venous malformation or familial venous malformation syndromes and others. In that case, the biallelic defect of an inhibitor of the pathway causes the increased signal flow. And this may be quite difficult to distinguish. In case one, I'm showing here, with hemihypertrophy of the right leg and a complex vascular malformation, we identified a KRAS mosaic mutation in the lesional biopsy. In this case, which looks quite similar with some overgrowth of the right leg and a complex malformation, vascular malformation and capillary malformations on the skin, the cause was a frameshift mutation that was in the germline and very likely an early second event in the affected tissue. This could be demonstrated in leukocyte DNA. So this shows us already the difficulties in genetic diagnosis. In an ideal world, in the case of a disseminated mosaic phenotype, you would expect that in every tissue you can obtain, you find a similar frequency of the mosaic mutation. But this is not true. Instead, there is often a very wide distribution of mosaicism among different tissues. And for some of these disorders I mentioned, especially the blood, the leukocytes, hematopoietic cell line shows a very low level of even absence of the mosaic mutation. In the case of a segmental mosaic phenotype, as we see in the oncogenic mutations, it is very often that the mutation is only found in the affected tissue, while unaffected tissue and also blood is negative for the mutation. So if we suspect a mosaic lesion or mosaicism in the tissue, we have to consider that the underlying or overlying tissue of the lesion contains different cell types and germ layers. And it could be that the distribution of the mutant cells is disseminated throughout all germinal layers, and every biopsy that you would take from this tissue would give you a small percentage or varying percentage of the mutation. In some other cases, you can find the mutation just in the tissue that is affected in different layers, but it can also be that it's restricted to one specific layer, like the epidermal cells, or in the case of vascular malformations, in endothelial cells only, while the surrounding tissues and cells do not contain the mutation. Two strategies can help to detect mosaic lesions. One is to use assays with increased sensitivity, and the other one is to use samples where we assume that the mutant allele is enriched. And in most cases, a combination of these two strategies have to be applied. Assay sensitivity can be increased by selective PCR methods, by digital PCR, such as digital droplet PCR, or ultra-deep NGS sequencing strategies. We will hear about that in one of the following talks. And the frequency or the content of the mutant allele can be increased by lesional biopsies, scratch biopsies in the case of epidermal nevi, or microdissection methods. It has to be pointed out that selective PCR methods and digital PCR are mutation specific, and only ultra-deep NGS sequencing technologies allow a broader screening for mutations in the sample. This is a case of a frontal arteriovenous malformation, and the mutation of MAP2K1 could be identified in a mosaic state with an allele frequency of 7% in a small biopsy from the lesion. We have shown previously that in the case of epidermal nevi and Proteus syndrome, a scratch biopsy from the epidermal nevus can efficiently demonstrate the mutation. A very new approach for mosaic detection is based on cell-free DNA. The promise of this approach is to confirm the tissue mosaicism in a routine blood sample. While leukocytes often do not contain the mutation in mosaic disorders, cell-free DNA is derived from various tissues and may show mosaic mutations. The feasibility of mosaic detection via cell-free circulating DNA has been shown in 2 recent papers. In one case, it has been shown that 3 of 5 patients with PROS disorders, CLOVES syndrome and similar disorders were positive in cell-free DNA for the PIK3CA mutation with an allele frequency that was quite low, ranging from 1 to 9%. In the case of vascular malformations, the detection rate was quite low for simple vascular malformations, and the allele frequencies also very low, ranging from 0.4 to 2.1%. In the case of lymphatic malformations, the cyst fluid seems to be a source that has a higher allele frequency for the mutation that may be up to 8% in this study. We have found in one case with caposiform lymphangiomatosis, a similar picture is taken from literature of this lesion, a complex mutation of the HRAS gene, which was present in 6% in the lesional biopsy, and we found the same mutation in 0.4% in the CF DNA using an NGS-based method Dennis Janse will explain to you in the next talk. Finally, I want to point out that the knowledge about the underlying genetic cause of a mosaic disorder, specifically in the case of vascular malformations, may have therapeutic implications as new therapeutic strategies are evolving that are using specific inhibitors, and therefore it is important to know where the dysregulated pathway signaling is taking place. With this, I want to finish and thank you for your attention.
Thank you for this very interesting presentation. We will see you later for the Q&A session. Let's move on to the second talk, Reconciling Regulation and Innovation with Standardized Software. For this, I would like to introduce to you all Dr. Ben Liesfeld, who holds a PhD in physics, worked some years at a startup for ophthalmological medical devices, where he played a key role in the development of computer-guided laser surgery devices. In 2013, he got first in contact with NGS diagnostics while working at a clinical laboratory, where his role was to establish IT, software development, and bioinformatics department. This inspired him to found his own business. Now he's the co-founder and managing director of Limbus Medical Technologies. Welcome, Ben.
Thank you, Yvonne, for the kind introduction. Genetic testing laboratories today must work within the tensions of regulation and amazing innovation in their field of medicine. And I would like to show how these tensions can be bridged by using a standardized software like varvis. Let me first remind you of the changes in the regulatory framework that are imminent for us. The transition period of IVDR will end next year in May of 2022, and from that moment on, medical devices can only be put into the market, placed into the market, and certified according to the new rules of IVDR. That means in particular that anything that is related to genetic testing will be classified as Class C, which is not a low-risk classification. This means in particular that Class C devices must undergo review by a notified body in order to demonstrate compliance with IVDR. Very important for laboratories in Europe is health institutions are not exempt from this regulation. Unlike in the United States, where laboratories are exempt from regulatory oversight, laboratories in Europe are not. They are rather required to use CE IVD products whenever possible, and if they intend to use research use only components, they must basically undergo a process that is very similar to a full CE approval. So what does constitute a laboratory developed test? Let us quickly review the standard process where a patient sees a physician, where the physician sends in a sample to a medical laboratory and the medical laboratory returns a report. In case that the medical laboratory used in its process a research use only component, it may itself become subject to the same requirements as a medical device manufacturer under IVDR. And these requirements are very prominently described in Article 5 of IVDR. Article 5 states in the very beginning that laboratory developed tests must comply with the general safety and performance requirements. And those requirements are very comprehensive. It also states that laboratory developed tests may not be passed on to other legal entities. And the laboratories must establish an appropriate quality management system, which typically means being compliant with ISO 13485. And in the end, laboratories must also establish a post-market surveillance process. They must monitor the analytical performance of their laboratory developed tests. So if you're now considering creating your own laboratory developed tests, you should pay attention to which components are you going to use for your internally developed assay. If you start with research use only components, they basically provide you with no level of documentation at all. When moving on to general laboratory use components or devices meant for performance evaluation studies, the level increases, but the highest level of documentation, of course, is provided by properly CE labeled medical devices. On top of the documentation that comes with a medical device, varvis also provides validation as a service, and this works as follows. The laboratory sequences appropriate reference samples, and those samples undergo the same process in the laboratory and the analysis in varvis as any other sample, and we compare the results from varvis with the published reference data. So in this way, the laboratory, the customer, receives a full validation report stating the analytical performance of the entire test platform. While this picture here describes a simple workflow for a germline assay, we also provide guidelines for more complex assays like liquid biopsy assays as described in the satellite meeting. Once the validation has been completed and the performance has been measured, specific quality criteria are defined and thresholds for quality metrics are defined. And these metrics can be monitored on several levels in varvis. The very basic level is a sequencing batch-oriented view on the data where issues in the wet lab can very easily be detected. There's another level, a case-specific level, where you can drill down from virtual panels down to genes or to the exon level and review quality-related data. But you also have quality metrics related to every single finding. And all these data are stored in the varvis database and can be monitored over time. And so we were very happy when the University of Magdeburg, the Institute of Genetics, approached us with their project to establish a high-precision liquid biopsy assay for a specific patient group. And we were happy to assist not only during the selection of a suitable technology, but also during performance evaluation and, most importantly, during optimization. That means providing not only the best performing assay for the patient, but also a cost-effective assay. Thank you.
As our final speaker today, I would like to introduce to you Dr. Denny Schanze. He has a background in molecular medicine and obtained his PhD in human genetics at the University of Erlangen-Nuremberg. Since 2010, he works as a scientist at the Institute of Human Genetics at the University Hospital Magdeburg. With his position now, he is the head of the array and NGS laboratory. He has already successfully established NGS panel and exome analysis in clinical routine in the past, and today he will tell us about his latest project related to NGS-based liquid biopsy. This talk is called Matching Analytical Performance and Clinical Utility. Welcome, Danny.
Thank you for the kind introduction and welcome all to my talk at the end of the session for matching analytical performance and clinical utility. Only a brief overview on how we use NGS sequencing in our lab. So as most of the others, we do a lot of whole exome sequencing. So for studying neurodevelopmental disorders, intellectual disability, and many other diseases. But we also use targeted panel, and we use them for also copy number analysis. And we know that the coverage is much higher than, and the analysis for copy number is much stable. But today I want to talk about a strongly focused mutation, so the hotspot panel. So I don't know whether you can see there is a small dot within this blue circle. It's really oversized if we think about the KB, the size of the custom panel we talk about today compared to the whole exome. But there is a small dot, and you will see hopefully at the end of my talk why it's necessary to focus on such a small panel if we think of liquid biopsy and study other mosaicisms or really targeted somatic panels. So maybe I will have 2 or 3 slides, so giving an introduction to the background that is necessary for my talk today. So if we think on sequencing strategies, so we need different applications. If you look for a germline mutation, so where we use large panels or whole exome sequencing, we look for heterozygous or homozygous variants. So therefore, the limit of detection is above 20 or 30%, is much easier to detect these variants also on a raw coverage about 80 to 150x. So we only need data application of the pooled samples to separate the reads between these samples. But if we look for a solid tumor or a somatic variants, so the limit of detection will drop to 5 or 1%. So therefore, we need a different approach, so called UMI technology. So the unique molecular identifiers and the raw coverage will increase dramatically. In the next slide, I will show you why this is the case. And also for liquid biopsy, maybe so the limit of detection further drops because the amount of DNA material we are interested in within the sample is quite low. So we have to focus on very small panels below 100 kb, maybe below 20 kb. I will show you later. And also the method that is needed for doing this analysis will change to duplex sequencing, and the raw coverage will further increase. So let's allow me to have a few words about the error rate. So we have to talk about that because we need the most accurate method to differentiate between the true positive variants and the false positive variants. And therefore, the error rate is very important. So if you have a read, let's say 100 base pair. So if we talk about an error rate of 1%, then in average, one base in this 100 base pairs will be an error in sequencing or in amplification error that rises up. So therefore, the coverage is really necessary. So therefore, the change from the germline analysis to the somatic or to the liquid biopsy, the need for more coverage is very, very important. Because if you have statistical in each of these reads one error, then it rises up in different positions. But if they overlap in one position, and we have 100 reads, and in 2 of them the error occurs on the same position, then it looks like a variant of 2%. And decide whether this is a true positive or a false positive variant is very critical. especially if we look for variants in the liquid biopsy below 1%. So therefore, we need a higher accuracy in noise reduction and in controlling the error rate. And how can we do this? So let's say we have here a small barcoded molecule with a true positive variant. So on the forward read in green marked and in the reverse read in blue marked. So this is the true positive variant. If you do the amplification of this strand separated reads, there occur with, let's say, 1% error rate, there occurs some artifacts on different position. If this occur later on in PCR amplification, there's really on different located on different positions. But if this occur in really early steps of the PCR amplification, maybe sometimes they occur on the same, because if it's incorporated in the early PCR steps, then they will be in all further reads or fragments that will be amplified from this. So now we can reduce only the forward strand and look for true positive variants here and the false positives and take them out. But in this case here, also the consensus strands for the reverse reads will rise up in this as a possible true positive variant. But if you combine these two strands, we will see that only this variant is a true positive, and therefore the high accuracy will be made. So if you took into account that we have in first here for our example and 1% error rate, therefore by using this separate consensus strands making, we can dramatically reduce this error rate and get at the end high accuracy reads with good differentiation between true positive and false positives. And we will see later on how this works. So at the beginning of our approaches or studies, we really ask ourselves the following questions that were very important. So is the single UMI technology sufficient to do this analysis, or do we need the duplex sequencing? What is the sensitivity if we think on the liquid biopsy, as I showed you in one of the first slides, that we want a level of detection of 0.5%. So what is the sensitivity? What's the precision of the assay? How much target region can we sequence? Can we do larger panels, or if you really have focused on a hotspot panel that is very small, and this is really important. And at the last, optimum compromises between clinical utility and the costs of all, yes, and the analytical performance. And also, therefore, I will show you some really exciting data that also addresses this. So what have we done? So we made two approaches. I call them approach A and B, and let's start with approach A. So therefore we use the single UMI, so-called unique molecular identifier technology from Agilent, also called the XTAHS1. So here on the next slide, you can see directly taken from the Agilent manual an overview on how does the library that are generated for NGS sequencing looks like. So in the middle in blue, we have our target flanked by some adapters. And on one side, we have the molecule barcode that is 10 nucleotides long. So if you combine them, we can take over 1 million different unique molecules directly in the beginning and differentiate them in the analysis process. On the other side, we have the normal sample barcode for demultiplexing and pooling some samples within a sequencing run. As Ben said in his talk, we also need a reference sample. So we used a commercial available reference sample that was validated by different technologies, which contains 15 spiked in hotspot mutation variants with a variant allele frequency on 0.5 or 1% respectively. So we ordered them, we sequenced them, and later on we analyzed them and looked whether we could identify these variants, if what's the false positive rate, was it the true positive rates, and all the other parameters we will talk about later on. So we designed a custom panel, and we started with a very small panel, so nearly 37 KB in size that targets 144 region with hotspots mutation that we are interested in and sequence them on an Illumina NextSeq. And first of all, before we go into the detailed analysis, let's have a look on the usable reads. So first of all, we decided we looked into full data sets, which are part of the reads we can really use. And therefore, we have to think about the unique molecular identifier technology is necessary to differentiate between the true positive and the false positives. Because if we see that a variant arises or is tagged with two different unique molecular identifiers, this means that from the beginning, this variant was detected on two different molecules and therefore is more likely a true positive variant than compared to reads with variants that only tagged with one unique molecular identifier. And therefore, only half of all our raw data really fulfill this criteria and were therefore usable reads. That 50% of all the reads we cannot use for our analysis. So for some reason, we make a bioinformatician subsampling because we are also interested in how many samples per run we can analyze, and to not do this in several runs, we choose a bioinformatician way to subsample this data set to only 50% and therefore save sequencing costs. And we identified in this subsample set with only 50% of the reads equally distributed or for all our target region, that the useful amount of reads drops quite below 50%. So that nearly 40% we lose another 10% of the reads, and therefore it's an indication that maybe the doubling of samples per sequencing run will not very useful for our analysis. So if you have a look on the coverage, so how does our samples and sequencing reads are equally distributed and on which coverage? As you maybe remind one of my first slides with the coverage for the whole exome sequencing, we have an average from around 100x. In the somatic, we need more than 2,000, and also in the liquid biopsy, maybe more than 10,000 raw reads. So this is our processed reads, and we see that in the full data sets, more than 96% or 96% reach this 2000x, and in the full data sets, 83% the 5000x. But in the subsampling, so the data for the 2000x seems quite similar to the full data set, but here in the 5000x, so the coverage drops quite down to 60%, and also therefore indicate that this isn't enough for the analysis. Let's have a look on the detailed calling performance. So in this sample, as I showed you in the beginning, we have 15 variants that were spiked in at 1% or 0.5%. And if we look on the true positive variants, so these are the good news, we detect all of this really true positive spike in variants. So we have a sensitivity of 100% even for the 0.5 level, also for the 1 point level of variant allele frequency. But the bad news are we had quite a lot of false positives. So we have additional variants that are not in this reference sample, in that really accuracy and validated sample. So these are also false positives. So the precision of the assay isn't good enough. And therefore, it's clear that at this stage, we didn't move on because we need a better noise reduction and therefore maybe another technology to do this analysis. And therefore, we switched in approach B to the duplex sequencing. So compared to the first approach, we also have on this library XTSH2 from Agilent the target, but now it's flanked by 2 times molecular barcodes. So each side, therefore duplex sequencing, and they doesn't have 10 nucleotides, so they are 2 times 3. So in combination, we can take more than 4,000 molecules, and we have also unique sample index on both sides. And therefore, we moved on with this. So we use the same reference samples, but by further reduction of target size, because we want to increase the raw data per sample, we only had 8 remaining of these mutations within our target regions, but at the same frequencies of 0.5 and 1%. And by reduction of target size, we only had 98 target regions with this hotspot mutation, and therefore overlapping with these 8 mutations in the reference sample, and the sequencing was done on the same Illumina NextSeq sequencer. So first of all, again, a look on the full data set and the usable reads, and by a reduction of the target size, so the custom channel was nearly half of the first one, we could increase the usable reads about 50, so it's nearly 58 or nearly 60%. So compared to approach A, we could slightly increase the usable reads, and that's a benefit for the analysis. But also have a look on the subsample data set. So again, bioinformatician subsampling of the reads to nearly 50% showed that we also comparable to approach A, we lose a lot of usable reads, that we have here only 40% compared to this nearly 60%. Therefore, indicating that maybe also in this approach, the increasing in samples per run will not work, but we will see. Also here, the comparison of the coverage. So we could slightly increase compared to approach A from 96 to 97% at the 2000x level, and from 83 to 85%. So the two approaches are nearly comparable. But if you have a look on the subsampling, so therefore the results are really dramatically drops down. So here in this of the 5000x level, we only have 33, so 1/3 of the sequences compared to 60% in approach A. But now have a more detailed look on the real data of the calling performance. So from the 8 true positive samples in this reference samples, we could all 8 true positive variants at a level of 0.5 and a level of 1% could be detectable. So the sensitivity of this assay and this approach will be 100%. And here, the further good news, we have no false positive, we have no false negative, so also the precision of this assay with these parameters, with a very low custom panel and a very high input read amount, is really good. So therefore, we got the best performance. Compared to the subsampling dataset with only 50% of the reads, so that was the simulation of doubling the samples per run. We see that also at the 1% level, we have the same results. But if we look on the 0.5 levels, we missed 1 true positive variance, what we are not interested in. And also we got 1 false positive. So this is a sequencing error that will be rise up as a true positive variance, but it isn't. So therefore, if we increase the sampling per run and have only 20 million reads as input for our analysis, then we must make compromises regarding the performance, what we are not interested in. So to summarize my talk, so we have to focus on really small target regions and using the duplex sequencing technology if we want to do the liquid biopsies. But also on this level, we have high sensitivity of 0.5 and a high precision regarding this large amount of input reads. And we are quite hopeful looking forward that this approach of this really read consuming assay and workaround will be suitable to study some liquid biopsies and some of the diseases we are interested in. And so far I presented a really newest data. We are looking to make some custom panels, so we have to decide not to make one larger panel, so we have to decide to make several small panels to reach this high input for data analysis. And therefore, we are looking forward to maybe present in one of the next webinars our newest information on further developing these tests on liquid biopsy samplings.
Okay, so thank you for these interesting talks. Now we have Professor Zenker and Ben Liesfeld here for the Q&A. So please feel free to ask your questions in the chat function if you have any questions. One question that we hear quite often is sorry. Does your software support barcodes from other manufacturers as well? So Ben, would you like to comment on that?
Yes, so the software is agnostic of kind of the technology or of the manufacturer. We support products from several different manufacturers. Sometimes when new products come out, there is some lead time involved, developing a proper workflow. But as of now, I would say we support all known products with respect to UMI in the market.
Okay. Would it not be beneficial to use barcodes also in NGS panels from germline diagnostic is another question.
We have received such questions before. Our customers have asked us to evaluate if barcodes provide a benefit also in their routine germline assays. And we evaluated this, and as you also heard in Danny's talk, it is rather important to achieve, in order to actually utilize the barcoding, is important to achieve a high duplicate rate. And typically in germline assays, you don't sequence with such a high depth of coverage, so that you don't actually have many copies of your reads in the data. And then it actually doesn't make sense, or there's no benefit of utilizing the barcodes. So we don't recommend to do that. It's just an overhead in computation.
Okay, and then the question back to the clinical perspective is, do you think that this assay is suitable to be performed in routine diagnostics? Maybe Professor Zenker, do you want to comment on that?
Yes, sure. I think there's a problem with mosaic diagnostic is, as I mentioned, is that we would like to find a method that is able to detect the mosaicism without taking a biopsy. And in some cases, it's really hard to take a biopsy. If you think, for example, of vascular malformations, so taking biopsies directly from the vascular malformation has bleeding risk, and so on and so on. So we really think that this is an improvement, and the need we have to consider is to be able to screen at least several hotspot mutations in one assay, which is hardly possible with ddPCR, because there are some multiplex ddPCRs that can tackle 4 or 5 mutations, but we can screen quite a large target region for this specific interest. And therefore, we think it is quite useful for that. We have also some experience, and as it can be anticipated, we see that we can find a mosaic mutation in the cfDNA if the amount of mutated cells has a critical size. So the very small vascular lesions will be really hard to detect, and this is also consistent with results in the paper I mentioned, where only 3 of 11 or 12 samples with simple vascular malformations had been detected by a cfDNA assay.
And from the perspective of the software and the assay, Ben, what do you think? Is it suitable for routine diagnostics?
We are listening very closely to what our customers are saying, and of course there are ideas how to further improve and streamline the analysis. From a bioinformatics point of view, I think we have everything ready and available, and we are happy to work with our customers to further develop the application to make it even easier to use in the routine diagnostics.
Okay. So at this point, we don't have any further questions. So I thank you all for your attention, for joining the Corporate Satellite today, and enjoy the conference. If you have any other questions that will come up at a later point, please feel free to contact us anytime. And so long. Take care. Goodbye.